Okay, so to round off our consideration of bacterial energy metabolism, I'm going to go through the different types of phototrophic bacteria and how photosynthesis contributes to their generation of metabolic energy. Now, the first thing everybody has to realize is that there are basically two independently evolved systems of photosynthesis in bacteria. So all photosynthetic eubacteria have a photosynthetic system based on bacterial chlorophyll, whereas the photosynthetic archaea that exist generate energy from light using a different um, chromophore, different light harvesting compound. So we'll talk about archaea first because it's basically the simplest. So archaea, photosynthetic archaea, generate energy from light based on bacterial rhodopsin. So bacterial rhodopsin is a 7-transmembrane helix protein, which basically functions as a proton pump. That is, the bacterial rhodopsin chromophore, which is buried inside the 7-transmembrane helix, once it absorbs light, this generates a kind of transfer across the membrane of one proton from the cytoplasmic phase to the periplasmic phase. So what is basically happening is that, light is used to generate a proton motive force across the plasma membrane. Now, there's no electron donor in this process and no electron acceptor. And what the proton motive force is going to be used for is, of course, to generate ATP using the proton ATPase. So these protons will, be retransported back into the cytoplasm, generating ATP during the process, and then they'll be repumped out into the periplasm by bacterial rhodopsin. Now, the bacterium which does this is Halobacterium salinarum. And you might ask, so what is the rest of its metabolism? So basically, Halobacterium salinarum is an aerobic chemoheterotroph. So it will use organic carbon as the basis of its energy metabolism by respiration. So you think, well, why is this thing performing photosynthesis? If it's a chemoheterotroph, it's going to be eating organic carbon. It doesn't need to generate energy from light. And that's true. In conditions of high oxygen, Halobacterium salinarum does not express bacterial rhodopsin and does not generate energy from light. However, under low oxygen conditions, when there's not enough oxygen for the bacterium to use respiration as its mechanism of generating metabolic energy, then bacterial rhodopsin will be expressed, and this allows the bacterium to generate more ATP, even in the absence of respiration. So if you remember, the main drawback or the main limitation of fermentation as a means of generating metabolic energy is that it doesn't generate enough ATP, okay? So that kind of helps understand what the role is of bacterial rhodopsin and this photosynthetic pathway in the metabolism of Halobacterium salinarum. It's there as a kind of means of making anaerobic metabolism more efficient. Okay, so that's the purpose of this photosynthetic mechanism in a bacterium, which is basically a chemoheterotroph. Okay, so that's for bacterial rhodopsin and photosynthetic archaebacteria. How about photosynthetic eubacteria? So, in fact, there are several types of photosynthetic eubacteria, and they all use a photosynthetic machinery based on bacterial chlorophyll as the chromophore. And when you look at the phylogenetic position of the different types of photosynthetic bacteria, you realize that they're all over the place, okay? Chloroflexi is up here in one of the basal taxa. Heliobacteria are gram-positive, and the other photosynthetic bacteria are all gram-negative, but they're kind of scattered around in different major groups of gram-negative bacteria. So the different types of photosynthetic eubacteria did not inherit their photosynthetic system based on bacteria chlorophyll from a common ancestor. They got the photosynthetic system by lateral gene transfer. That's an important message. And that's why you find photosynthetic bacteria in different bacterial phyla. Okay, so in eubacteria, photosynthetic eubacteria, most of the groups only use one reaction center in their photosynthesis, whereas cyanobacteria, just like chloroplasts, they have both reaction centers, and they have photosystem 1 and photosystem 2. And it's not really obvious why, some photosynthetic bacteria have managed to acquire photosystem 1 but not photosystem 2 during these lateral gene transfer events. But the model, which was published more than 15 years ago now, posits or supposes that initially you have some type of bacterium which is the ancestral photosynthetic eubacterium, which they call the procyanobacteria here. And so this bacterium evolved first photosystem 1 because the proteins apparently involved in this pathway seem to be more archaic than those involved in photosystem 2. And then by some gene duplication event evolved also photosystem 2. And that this bacterium would be using both of those photosystems, not at the same time, but differentially depending on the environmental conditions. And then from this kind of like ancestral photosynthetic bacterium, different lateral gene transfer events would transfer either photosystem 1, reaction center 1, or photosystem 2, reaction center 2, to different groups of photosynthetic bacteria. And the last step in the evolution of photosynthesis among bacteria is the kind of integration of these two different photosystems within cyanobacteria to allow oxygenic photosynthesis to occur. So what I'm going to do now is talk about one type of bacterium which utilizes reaction center 1, photosystem 1, that is chlorobi, the green sulfur bacterium. And then for an example of photosystem 2 operating on its own, the purple non-sulfur bacteria. Okay, so green sulfur bacteria, for example, chlorobi, they have photosystem 1, and its function here is to take electrons from a reduced sulfur compound like hydrogen sulfide, and then the absorption of light, so this electron will be transferred to the base of photosystem 1, and then the redox potential of this electron will be boosted by the absorption of one photon. Then what happens to this electron? Well, then it gets transferred, or the pair of electrons get transferred down an electron transport chain, and finally end up reducing NAD plus to NADH. So how is this fitting in to the basic type of energy metabolism of these bacteria? So in this sense, green sulfur bacteria are basically lithotrophs. Okay, they're using an inorganic source of an inorganic molecule as a source of electrons and electrons. And so The function here of the photosynthetic part is to boost or increase the amount of energy that can be extracted from this redox reaction. So what is happening is that light in the photosynthetic reaction here is increasing the redox potential of the um, half reaction of the oxidation of sulfide to molecular sulfur. Now, if you remember, one of the drawbacks in or for sulfur oxidizers among the chemolithoautotrophs was that the redox potential of oxidation of sulfur is not enough, of, or sulfide rather, is not high enough to transfer electrons directly to NAD+. So in order for chemolithoautotrophs, sulfur oxidizers, to generate NADH, what they have to do is use up some of the proton gradient that they generate from this from ATP to push back electrons onto NAD+. So for green sulfur bacteria, the photosynthetic green sulfur-based lithoautotrophs, the advantage of having the photosynthetic system is that NADH can be generated directly. And that means that the proton motive force that's generated also by this redox reaction can be used exclusively for the generation of ATP. So it's a real gain in efficiency of the redox reaction, which is at the base of the metabolism of these bacteria. So of course, this type of photosynthetic metabolism requires a good electron donor, okay, which would otherwise just be used by chemolithoautotroph. So you might think, well, okay, so if this is so great, why aren't all the chemolithoautotrophs, why aren't they all photosynthetic? Okay, well, one of the reasons is they don't all live at the surface of aquatic systems where there is access to light. So if you don't have any light and you're a chemolithoautotroph, well, you can't be photosynthetic, of course. Okay, so that's photosystem 1, a type 1 reaction center acting on its own to kind of boost the efficiency of our metabolism, which is otherwise lithoautotrophic. Okay, how about photosystem 2 working on its own? This is what you find in the purple non-sulfur bacteria. So here what is going to happen, like for example, Rhodobacter. So here what is going to happen is that light is absorbed by the type 2 reaction center. The electrons, this will boost electrons up, and then afterwards they're going to be transported down an electron transport chain, and the electrons cycle around, okay? They kind of slide down the electron transport chain and they feed in back into the base of the reaction center. And then they're boosted up again by absorbing light. So what is the purpose of all this, these electrons going round and round and round? Well, the important thing here is you've got this quinone molecule in the electron transport chain. So every time quinone picks up 2 electrons, it will also pick up 2 protons from the cytoplasmic face of the plasma membrane. And when it gives up these electrons to the cytochromes here, which will then feed into the base of the reaction center, the photosynthetic reaction center, then the 2 protons that are carried on the quinol are released in the periplasmic phase of the membrane. So every time electrons cycle around here, then it will generate a proton gradient because the quinone is picking up electrons on the cytoplasmic face of the membrane and releasing them, picking up protons on the cytoplasmic face of the plasma membrane and releasing them on the periplasmic face. So That means that the light is being, the energy in the light here is used to create a proton motive force, which can then be used to generate ATP. Okay, and so there's no electron donor here, and the electrons are not passed on to NADH. So what is the basic metabolism of these purple non-sulfur bacteria like Rhodobacter? Well, they are basically anaerobic chemoheterotrophs. So they're using organic carbon as a source of energy and a source of electrons, but they don't have access to oxygen. So they do not perform anaerobic respiration either. So their energy metabolism from their source of carbon is a little bit inefficient. And as I said before, just like for Halobacterium salinarum, the archibacterium that uses bacterial rhodopsin, the drawback that they have, or the limitation that they have in their energy metabolism, is that anaerobic fermentation pathways, for example, are not generating enough ATP. So for purple non-sulfur bacteria in an anaerobic environment, like if they have light, then they will express the proteins involved in this type 2 photosynthetic center, and the light energy allows them to generate a proton motive force and more ATP, which they can't otherwise do because they do not perform respiration. So once again, these bacteria basically they've got some other kind of energy metabolism, and photosynthesis is just added on top. Now, the final example is cyanobacteria. So these are the only photosynthetic bacteria that have both type 1 and type 2 reaction centers. And cyanobacteria perform oxygenic photosynthesis just in exactly the same way as chloroplasts in all green plants. And of course, cyanobacteria are basically the bacteria that were internalized by green plants in an endosymbiotic event. So what is happening here is you have the two photosystems working one after the other. So in the beginning, light is, well, it's not exactly the light energy, but basically an oxygen evolving complex will remove 4 electrons from 2 molecules of water to produce a molecule of oxygen and 4 protons. These electrons get passed on to the reaction center of photosystem 2, boosted up by absorbing light, and then they are transferred down the electron transport chain onto quinone, because this is basically a photosystem 2 system here, okay? But instead of these electrons being transported back down to the first photosystem, they're transported down to photosystem 1. And from here, they get boosted up again, and then another electron transport chain will end up transferring them to NADP plus to give reduced NADPH. So here you have both things working in parallel. So you've got an electron donor all the way over here, which actually is a very, very poor electron donor. But the fact that you've got these two photosystems working in parallel generates enough redox potential to transfer these electrons onto NADP+. So you can get NADPH or NADH generated directly. And of course, at the same time, you'll generate a proton motive force because you've got the quinone cycle, which is occurring here. So every time you get these quinone molecules transferring electrons, they're also transferring protons from one side of the membrane to the other. And this proton motive force is what allows cyanobacteria to generate ATP. So basically, these bacteria, of course, can live on almost nothing, okay? They get metabolic energy just from water and sunlight. That's it. So, of course, we require an electron donor right at the beginning of the process, but water is ubiquitous. So this is an incredible evolutionary advantage, which means that, cyanobacteria are by far the most prevalent and the most diverse group of photosynthetic bacteria that exist on the planet. Okay, so that's for bacterial photosynthesis. So this is just a summary table here to show you the different types of photosynthetic bacteria: green and purple, sulfur and non-sulfur bacteria, and cyanobacteria. So chloro-B, green sulfur bacteria, reaction center 1. They use reduced sulfur compounds as the electron donors. They're basically lithoautotrophs. They're anaerobic, and so light is used to increase the efficiency of their lithoautotrophic metabolism. So that's kind of the same for the purple sulfur bacteria, although they use a different photosystem to do the same thing. Chloroflexus, the green non-sulfur photosynthetic bacteria, and purple non-sulfur bacteria. Now they're basically chemoheterotrophs. They require a source of organic carbon as their source of electrons, and for their source of carbon for building structures. And here they are anaerobes, so the function of the photosynthetic system is to increase the efficiency of their anaerobic energy metabolism. And cyanobacteria, they are both photosystems. They use water as the electron donor, they fix carbon, and they are the only oxygenic photosynthetic bacteria. Okay, so that's it for photosynthetic bacteria, and the last summary slide of the different types of energy metabolism that bacteria perform. So in respiration, basically the electrons are, or the energy metabolism works like this. So the source of everything is an organic source of carbon. Its oxidation will result in the reduction of NAD plus to NADH, which then will be NADH will then give up electrons to an electron transport chain, which finally transfers the electrons onto oxygen or another good electron acceptor. During this or electron transport, bacteria generate a proton motive force across the plasma membrane, and the proton motive force is then used to generate ATP. So why is this useful? Well, it's because you get much more ATP per molecule of carbon than is possible to generate by fermentation. And you have to remember the difference between anaerobic respiration and fermentation. In anaerobic respiration, okay, bacteria don't use oxygen, that's why it's anaerobic, but they do require another external electron acceptor, like nitrate or thiosulfate. Whereas in fermentation, there's no external electron acceptor. The electron acceptor is pyruvate, which is itself derived from the source of organic carbon, which is at the base of the bacterial metabolism. Okay, for chemolithotrophs, where does the energy come from? Well, it's basically coming from a favorable redox reaction, okay, a chemical oxidoreduction reaction. So oxidation reduction, this is a transfer of electrons. So the base of the metabolism is redox reaction, which will feed electrons into an electron transport chain. So this electron transport chain can be used to, is used to generate, on the one hand, a proton motive force, which allows bacteria to generate ATP, and it can also be used to push electrons onto NAD plus to generate NADH, and this NADH will supply the reducing power required to fix carbon dioxide into organic carbon. So you see like the elements in the process are kind of the same, but the direction here is reversed compared to respiration. So, one of the things that's important is why only certain electron donor acceptor couples can work, okay? So in order for the basic chemical reaction to be favorable, you need the transfer of electrons from a good electron donor to a good electron acceptor. So good electron donors, molecular hydrogen, definitely the best, then reduced sulfur compounds, then reduced nitrogen compounds. Okay, and finally, phototrophs. What the things are to remember is, the polyphyletic nature of different photosynthetic bacteria, which means that these different groups of photosynthetic bacteria have acquired the machinery necessary for harvesting light and using it to generate metabolic energy by lateral gene transfer. They have not acquired these photosynthetic systems by inheritance from a common ancestor. And I think, when you think about the evolution of these photosynthetic systems, it seems like basically you would have functioning bacteria that have a different type of metabolism pre-existing, like, the green sulfur bacteria. One can easily imagine that these were basically sulfur oxidizing chemolithoautotrophs. And then they boosted their metabolism by acquiring photosystem 1. And it's the same for the purple and green non-sulfur bacteria. These are basically heterotrophs, and they boost their energy metabolism by acquiring the capacity for some type of photosynthesis. Okay, so that's the end of the 2 lectures on bacterial energy metabolism, and I'll see if this recording has actually worked. Otherwise, I'm going to have to record the whole thing again.